Thank you very much. So there really is a connection between diagnostics and therapeutics after all. So that is the last talk in our understanding biological activity to inform drug development. Now we'll be shifting to the second portion of our session, assessing early signals of efficacy to guide clinical development. And our first speaker is David Feltquate. He's head of early clinical development at Bristol-Myers Squibb, and someone who has maybe more extensive experience with the IO clinical development than many others. So David, welcome.
Thank you, Mace, and thanks to the conference organizers for giving me this opportunity to speak. I think Keith said it best earlier that as the meeting goes on, we'll probably see more and more of similar cases, and we probably won't have to emphasize certain parts of our talk. And I've found already now with about 6 talks down, about half of what I'm going to talk about has already been described. But it's perfect actually because it sets up a prelude for some of the background, and we can talk more about what the implications are. So what I wanted to do was walk through what it's like for someone in pharma as they're faced with looking at clinical data and having to make decisions around moving trials forward or moving drugs forward, expanding trials, stopping trials, and what that looks like. What are these decisions that we have to make? And so I decided to do this in terms of case studies, seeing what are the common themes and things that we could call out that might help guide how we make these decisions. And through these case studies, I hope to lay out what I want to be a little provocative and talk about what I think the future is going to be, because I don't think it's really around monotherapy and monotherapy decisions anymore. I think this is going to be about how we approach the development of these drugs in terms of combinations, triplets, really complex regimens. So I'm going to start off, we've talked a bit about PD-1 inhibitors already today. I want to start this off and really, again, remind everyone, go back in time where we were back, actually it was 10 years ago last month that the first patients were treated with PD-1 inhibitors. And I'm going to show you a little bit of data from that first trial a little bit later. But what I'm showing you here is some of the data from the first multi-ascending dose study, which began in late 2008. We were faced with something very interesting. So at this time, ipilimumab had not been approved. Ipilimumab was showing signals of activity as a single agent in melanoma. A little bit of data in renal cell. The activity of ipilimumab was single digit, maybe 10 to 15% response rates. And it was in tumors that were, quote, immunogenic. These were tumors we expected immune therapies to have activity. So what was really interesting in this study, this is called study 3, was we started to see, we did a dose escalation, and then we did an expansion in 5 tumor types. Those were melanoma, renal, non-small cell lung cancer, prostate cancer, and colorectal cancer. And what was remarkable was that 3 of those 5 tumors, we started to see initial activity. Now, this was small sample sizes. These were expansion cohorts of roughly 20 patients. But the thing that, the signal, the hallmark, I think that really called out that we had something special here was 2 things. One is a variety of tumors we were seeing activity, but most notably, it was non-small cell lung cancer. Showing activity with an immune therapy in renal and melanoma is important, but it's not the clue that you had something different. The other is as we amended the study, and as Eric has described for us, and Mace has described for us, this was an opportunity to amend a study and turn it into something much larger. And we expanded across multiple tumor types, and we were able to gain greater confidence that this was a real signal. And so in this case, we were able to see response rates of around 20 to 30% in these 3 tumor types. These responses now, over time, we're able to show were durable, and we were able to show other features of important clinical activity, which is shown in the bottom right panel, which is pseudoprogression. So these are progressors, these are not responders, but these are additional evidence of benefit. As a result of all that, we were able to do what at that time was considered bold, which was to take a new drug and go right from phase 1 into large randomized studies in multiple tumor types simultaneously. It was successful, and it was successful for a lot of different companies, a number of different agents. It's actually quite remarkable when you think about where this field has come in this amount of time. What I've put on here is a list of the agents that have shown or confirmed benefit in the context of randomized studies. And I've also shown agents that have shown evidence of clinical benefit either in single-arm studies that were designated as breakthrough by the FDA or approved by the FDA. So the evidence is substantial that even in this single-arm data that this is meaningful benefit. And I'm sure we're not over. I'm sure this list will continue to grow. So I'm gonna come back to one part. I want to talk about another example of a big signal. Right? When it's a big signal, it's very obvious. I think the decisions to move forward are easy. It's really around how aggressive you want to be at that point. I mentioned to you that it was 10 years ago that the first anti-PD-1 patients were treated, and it was in the context of a single ascending dose study, study 1. Now, some of you may ask, well, why was it a single ascending dose study? And this is an example of what I would call negative serendipity. So you might recall in January of 2006, there was a horrible experience. There was a company called T. Gennaro that had embarked on a normal healthy volunteer study with a CD28 superagonist. There were, I think it was 6 or 7 patients that were dosed within 1 hour of each other. They were all hospitalized and they were on the brink of death. It caused at that time a lot of fear and concern, rightfully so, about the use of immunotherapies and unleashing the immune system. So Medarex, in fact, I think Izzy Lowy is in the room right here, who drove this. He had to, they submitted an IND in April of that year, and there was pushback to turn that multi-ascending dose study into a single ascending dose study. Nonetheless, that single ascending dose study went forward, and something very interesting was found. There were 3 responses in the cohort of 39 patients. One with a patient with melanoma, one in a patient with renal cancer, and one in a patient with colorectal cancer. Now, these patients, once they showed benefit, were able to get subsequent treatments on a monthly basis. So this wasn't based on just one treatment. But what was remarkable in that colorectal cancer patient was this is a complete response. And I think, Suzanne, correct me if I'm wrong, I think this patient is still without disease at this point. So now at that time, in 2008, 2009, our ability to do robust sequencing was limited. I think at that point, what did it cost? About $100,000 to sequence a genome. So it was only a little while later that we started to understand fully what was the mutational burden in patients who have microsatellite instability or mismatch repair. But the group at Johns Hopkins astutely looked at this patient, determined that this was a patient who had MSI high, and that set up a hypothesis to do more focused prospective work in that subpopulation. Because this was also a study initially sponsored by nivolumab, we were discussing with them, and there were actually 2 studies that were initiated. There was an IST conducted by Hopkins showing robust clinical activity in the population of colorectal cancer patients with MSI high, but very little activity in patients with MSS. And likewise, BMS had conducted a similar study with nivolumab. So again, one patient, you can call it an anecdote, but there was good science tied into this, and that led to doing prospective work showing a very large signal. And as a result of that, Merck with pembrolizumab has breakthrough status with this, and they're in the midst of a review, and hopefully this will be approved soon to help patients with these subsets of disease. So I'm going to go back. So Ken uses as an example some of the work in myeloma, and so I'm not going to go into a lot of the details. He did a great job of providing this information. What I want to do, though, is have us think about this is really the situation we may be facing as we move forward. It's what do you do with a so-called false negative signal when you're not seeing robust activity with a single agent, new molecular entity? In this case, I actually have 2 examples. One of them is elotuzumab. Can describe this. This is an anti-SLAMF7 antibody. It has 2 potential mechanisms of action. It can be bound to NK cells and further activate them. It can also bind to myeloma cells that express this and make them available to ADCC. As a single, we hypothesized that from preclinical work, we expected to see some activity even with a single agent. We did not see that. So in the phase 1 study, there was very little activity and all the way up to the highest doses that were evaluated. There was some evidence of maybe some activity with stable disease and some minor reductions in M proteins, but this was nothing to write home about. And Ken described earlier, this is something that you might have just walked away from. But it was understood even early in the program that this is something that you might want to combine with other agents. And so that was done. And in phase 1, when 2 studies, elotuzumab was combined with lenalidomide. It was also combined with bortezomib. What I'm showing here is the data with lenalidomide. And this is in relapsed refractory patients with at least 1 prior treatment. And it's quite a robust activity. This then led to having confidence to do the definitive study, the confirmatory study, which was called ELOQUENT-2. And it was able to confirm through the endpoints of progression-free survival and with response rate that this was meaningful clinical improvements over lenalidomide dexamethasone alone. So the other, quote, false negative signal, Ken mentioned that, is with PD-1 inhibitors. So with nivolumab, there was a large phase 1 study in a variety of heme malignancies, a heme basket study. There were 5 types of malignancies that were evaluated. This was actually a study where we saw the initial signal with Hodgkin's disease. We also saw in T-cell lymphomas, follicular and DLBCL, some evidence of activity as measured by responses. Where we did not see very much activity at all was with nivolumab in myeloma patients. And so the next good study was done, and this was done with Merck sponsorship with pembrolizumab and with both pomalidomide, and there was also a study with lenalidomide. And what we can see here is that there is robust activity of this combination. Ken walked us through the biologic rationale for why that may be. What I've shown on the bottom is the PomDex data from the registrational trial for PomDex. And you can see here we're doubling the response rate. And I'm emphasizing the doubling because I think this is where it's really important we start to be able to, if we can, predefine, but to know what is a meaningful signal. And in this disease, I think this is quite meaningful. It's also notable that in the subpopulations that are double refractory or have high-risk cytogenetics, we're also seeing this kind of comparable activity. This is a meaningful signal. And as a result, that has spawned the investment in a number of confirmatory trials. So there's trials going on right now combining with pembrolizumab. There's a trial going on combining pomalidomide with nivolumab. We've taken another step in that randomized study. We've actually added a 3rd arm to it, which is an exploratory arm, actually looking at the quad of nivolumab, elotuzumab, Revlimid, and dexamethasone, the so-called NERD regimen, to see if we can actually capitalize on these multiple different mechanisms at once to further enhance activity. So that brings us to combinations. And this is where I think we need to spend a lot of time thinking about how we're going to know about good signals. So one of the combinations we've evaluated is a combination of IPI and Nivo. So ipilimumab, an anti-CTLA-4 antagonist, had already demonstrated clinical benefit in melanoma, had obtained FDA and EMA approval as a single agent in this disease. So we spent a fair amount of time trying to combine the two, and I won't go through the challenges of finding the optimal doses of these kinds of combinations. For melanoma, though, what we did see, which is shown in the lower left panel, is what we felt was robust clinical activity. So this is meaningful improvements in response rate, and I think more importantly in the depth of response. We were getting more CRs and near CRs with this combination than we saw with nivolumab alone, and certainly more than what we saw with ipilimumab alone. And that set us to make the decision to move forward and do multiple confirmatory trials to show the benefit. And result of the 3-arm study looking at each of the agents plus the combination of agents is shown here. Recapitulating, very similar to what we're actually seeing here with the waterfall plot and the degree of responses and tumor reduction. So what I'm going to leave you with is some examples of where we don't have the confirmatory data to know if these are meaningful signals. But I'm going to leave it up to your imagination. I can tell you that BMS has felt these are, and we've taken these steps to do the confirmatory work. So one of them centers around also ipi-nivo. In this case, it's the combination applied to first-line non-small cell lung cancer. Here, there's a different dose schedule, the combination of ipi-nivo for lung cancer. And overall, compared to nivolumab as a monotherapy, there appears to be almost a doubling of the response rate. When you break it out by PD-L1 positive, we're seeing there's a trend for more responses in the negative, but where we're really seeing the greatest numerical benefit and proportional benefit, a doubling of these responses is in the PD-L1 positive population. And that extends through the spectrum of different cutoffs of PD-L1 positive. Well, what's notable is, and very, very small sample size here, but we're starting to reach a point, as Ken was describing for myeloma, we may be starting to see this for other combinations where we're going to get to maybe near 100% response rates, and hopefully adding on other regimens or selecting the right subpopulations, we can turn these into perhaps complete responses. The last one is now it's the next generation of IO therapies. So this is data from a combination of nivolumab with something called lirilumab. It's an anti-KIR antibody. This is a monoclonal antibody that binds to KIR and it blocks KIR interactions which inhibit the activity of NK cells. So the hypothesis is that you combine these two together and you may see greater activity. I can tell you, I'm not showing the data, monotherapy lirilumab has very little activity. Mark Retain was describing earlier that you might want to dose up to toxicity, and that at least gives you some on-target toxicity, and that at least gives you some evidence that you're hitting the target and having an effect. With this drug, it was very, very little toxicity, and other than receptor occupancy and some minor pharmacodynamic data, it was hard to know whether you're truly having some impact. The combination of these two is quite remarkable in head and neck cancer. We know from a randomized phase 3 study of nivolumab in prior treated patients with head and neck cancer, you get a single agent response rate on the order of about 13%. And if you look at the biomarker positive population, it's very similar, it's about 17%. Whereas when you look at the combination, the early data suggests you're getting greater activity of the combination, but specifically it seems to be focused primarily in the biomarker, the PD-L1 positive population, where we're getting approximately a doubling of response rates. So the question is, small sample size, the question is this a real signal? Is this something that should be moved to randomized studies? What is the design of those studies? Is this a randomized phase 3, a randomized expansion? And this is where we're at right now. I mention this because the preliminary data of a number of IO therapies that have been presented at the last year and a half, 2 years worth of meetings, I don't think has been that impressive. It's been overall populations. It's part of the dose escalation. We're not honing in, but we're not seeing the home runs that we had seen with these initial agents. And what that tells me is one, we need to make sure we're actually hitting the targets and that they're having effects. But if they are, we can't just, we have to be careful we don't throw them away, and that we may be in a situation of what I would call necessary but not sufficient. And it's going to be important for us to find a systematic way to evaluate that, but to do it in an efficient way so we're not minimizing patients and how we use them in these trials. So just in conclusion, as we move toward this seamless development process, this faster process, I think we have to have some caution. And as we move these forward, especially as we move towards combinations, that we are seeing meaningful clinical signals. The hallmarks of these are going to be probably things like doubling of response rates relative to a control. Ideally, an inpatient control. I showed you historical data, but I think where we're moving towards is the in-trial controls. It's going to be maybe changes in the depth of responses built in with some pharmacodynamic evidence of activity. And then we need to still apply this kind of rigor as we move these new agents forward. Thank you.